Alright, so now in this part of the digestive system chapter, we will learn about the esophagus, the stomach, the pancreas, and the liver. And so here, this is talking about the esophagus and how it is the food passageway from the pharynx to the stomach, and it contains a lot of mucus glands. Then the mucus is making it easier for the food to go down the esophagus. The lower esophageal or the cardiac sphincter is the entrance into the stomach. And so I'm going to write this down for you. Entrance to stomach. So now the stomach. Stomach is the one that receives the food from the esophagus and mixes the food with the gastric juice. And here in the stomach, it's the proteins that are being broken down. So in the mouth, through saliva, it was starch. Now the stomach is doing the proteins. Has limited absorption. So here the stomach really absorbs more of liquids, medicines, medications, alcohol. Not much of nutrients of absorption occurs here. But then it's going to move the food next into the small intestine. The stomach contains these layers of smooth muscle. Know that there is a circular layer. This is more on the inner part. And there's a longitudinal layer. So both of these are muscular layers. Then there's a third innermost layer of oblique muscles. And so this is what they're trying to show here, the oblique layer of the smooth muscle layer. This is the innermost part. So then next there's a longitudinal, and then the circular layer is then the outermost. And so we're going to go over the parts of the stomach in the lab. Before lecture, we need to know what is inside this gastric juice. So here we're going to put a big star. Pepsinogen. Pepsinogen made by the stomach cells. This is the inactive form of pepsin. So know that it's released by these cells called the chief cells in our stomach. So then the pepsinogen, so this is the inactive form. It then turns into the active form pepsin. So know that this is the active form. And so know that the pepsin then breaks down the proteins. And so it's able to turn from pepsinogen to pepsin in the presence of hydrochloric acid. Know that hydrochloric acid is made by the parietal cells. And so again, this converts the pepsinogen to pepsin. Mucus, there are also these mucus cells in our stomach, so as well as in the esophagus. And they're making the mucus that allows for the easier movement of food, but also it's helping to protect the stomach lining. Then the intrinsic factor is also released by the parietal cells. This is for the absorption of vitamin B12. So here we're seeing the inner lining of the stomach. Just like with the intestines, there are these finger-like projections. So these are called the villi. And so along the villi, and also know that this is the mucosal layer, mucosa. And so here we have the mucus cells. There are the parietal cells, the chief cells. And so these are just then releasing the different enzymes. The regulation of the stomach is by nerve signals and also by hormones. And so you should know this by now. Sympathetic nervous system decreases digestive activity. Parasympathetic instead increases. Parasympathetic signals also increase histamine, which increase the gastric secretion. Okay, so then know here these hormones. Somatostatin released by the pancreas inhibits the secretion. Gastrin, this one increases. And then the CCK, also released by the small intestine, decreases the motility. So here, somatostatin and cholecystokinin. So here in this picture, what this shows is the parasympathetic signals going to the stomach to increase the stomach's activity. The stomach releases gastrin, and then gastrin stimulates the glands to release more gastric juice. And so here, gastric enzyme pepsin, we already said, know that it breaks down proteins. But so other nutrients are not broken down and also not really absorbed. But so here, some of the things that the stomach does absorb: water, salts, drugs such as aspirin, and alcohol. And so here now we can see how the stomach churns the food, mixes it in. Mixing of the food in the stomach then produces chyme. So think of this as the food mixed with the acid. Now we call that the chyme. And the peristaltic waves are pushing the chyme toward the end part of the stomach. And the end part of the stomach is called the pylorus. So then small amounts of the chyme is sent into the small intestine at a time. So here the pyloric sphincter is the exit out of the stomach going into the small intestine. But yeah, so it opens only in brief amounts of time because you don't want to send this acidic chyme into the small intestine. Small intestine is not designed to withstand the acidity, so only sending little amounts at a time will help to protect the lining of the intestine. And so this emptying of the stomach is regulated because of the sensitivity of the intestine. So this is called the enterogastric reflex. What happens is if too much is going into the small intestine, then a signal is sent to the stomach to slow down its emptying. Next, we're going to talk a little bit about the pancreas. So the pancreas is below and a little behind of the stomach. Pancreas makes insulin, glucagon, as we learned, but it also makes the digestive enzymes. We're going to learn that the pancreas makes any, an enzyme for any type of nutrient to be broken down. So it makes enzymes that break down proteins, it makes enzymes that break down the starches that were not able to get broken down in the mouth. It makes enzymes that break down lipids, nucleic acids. And so here we're going to go over the parts of it. And so this is the end part is called the tail, then the main part is the body, and the part where it's closer to the small intestine is called the head of the pancreas. And then we're going to see that here the bile duct coming from the gallbladder and the pancreatic duct are both opening into the small intestine in the same region. So they call this the hepatopancreatic sphincter for the gate, and the region is called the ampulla. Okay, so now next, the pancreatic juice. So as I was saying, pancreas makes these enzymes that can break down carbohydrates, fats, proteins, nucleic acids. And so here are some examples. You don't need to memorize these, but just kind of put these in here just to show you what these enzymes break down. Now the pancreatic secretion needs to be regulated. So here what it shows is the acidic chyme going into the duodenum, which is the first part of the small intestine. So then when the food, or I should say the chyme goes in the small intestine, intestinal mucosa releases secretin. Secretin goes into the bloodstream first, then secretin goes back to the pancreas to tell the pancreas to release bicarbonate ions. So the bicarbonate ions, these are a base, alkaline, so opposite of acidic. So now the pancreatic juices contain more bicarbonate ions, and this is to neutralize the acidic chyme that is going into the small intestine. After we eat, the hormones that are getting released to break down the food. So secretin, we already saw in the last slide in the picture. So this one stimulates the pancreas to release bicarbonate ions, so the base. And then there's also CCK. This one is released by the small intestine. But so then this one stimulates the pancreas to release pancreatic juices. Next, the liver. Liver, we're going to learn that it has the 4 parts to it. There's the right lobe is the largest, then next to that left. Then in the back, we're going to see a square-shaped part. That's the quadrate lobe. So quad, think 4, has the 4 sides. And the caudate is the smaller lobe, right next to the inferior vena cava. In between the right and the left lobe, there's the falciform ligament. And the bottom part is the round ligament. This is to attach the liver to the front wall of your abdominal cavity. When we look inside the liver, we see these shapes, these octagons, or hexagons, I'm sorry. And so these are called the hepatic lobules. In here are the hepatic cells, meaning the liver cells. And there are also a lot of blood vessels here. And so these blood vessels are bringing the nutrients absorbed from our digestive tract, and then the hepatic cells are going to process the nutrients. So we learned about the hepatic portal vein, and we learned about how the veins that are bringing the nutrients are connecting to the hepatic portal vein, and the hepatic portal vein is the one connecting to the liver. And so again, know that hepatocytes are the liver cells. These are the ones that are actually processing the nutrients. And also know bile is produced by the hepatocytes. Okay, so this is kind of a misconception. People think that bile is made by the gallbladder. No, so we're going to learn that the liver makes the bile and then gallbladder stores it. So here we're seeing the functions of the liver. One function, carbohydrate metabolism, so it's able to break down carbohydrates. It can even store carbohydrates within itself, within the liver. Lipid metabolism, it can break them down, turn them into different molecules if those molecules are deficient. Protein metabolism, liver is able to store vitamins, glycogen. Blood filtering as well, it's able to remove those old red blood cells, damaged red blood cells, and also any foreign substances. Detoxification removes the toxins from the blood, and the secretion part, it produces and then sends the bile to the gallbladder. But if the gallbladder is removed, then the bile just goes directly to the small intestine. Doesn't stop at the gallbladder. Replacing the liver is required to maintain life. It is actually able to regenerate on its own, but if there is cancer and it spreads to the liver, then it's not able to regenerate from that. But so donations can be made. Just only a part of the liver can be donated because regeneration of that part of the liver can actually help to fix the liver. There are these other devices that help to kind of mimic what the liver is doing.